Enzymes. Enzymes are proteins which act as biological catalysts. A catalyst alters the rate of a reaction without itself being changed. Research indicates that enzymes combine with the molecules they act upon, called substrates, to form an enzyme-substrate complex. The reaction that takes place results in products from the original molecule but leaves the enzyme unchanged. Enzyme-controlled reactions can be either anabolic, building up larger molecules, or catabolic, breaking down large molecules into smaller molecules. Enzyme reaction. Enzymes are protein molecules which are usually larger than the substrate. This means that only a small part of the enzyme, called the active site, comes into contact with the substrate. Fisher's hypothesis states that the active site has a special shape, like a lock, into which the substrate fits like a key. Factors affecting enzyme action. 1. Inhibitors. Enzymes have only one specific substrate. This helps to explain the lock and key hypothesis. If the substrate does not have the right shape, it will not fit into the active site. Enzyme activity can be inhibited by molecules having a similar shape to their specific substrate. The inhibitor blocks the active site and prevents the enzyme working. For example, sulfonamide drugs inhibit enzymes in pathogenic bacteria. 2. Temperature. An increase in temperature results in an increased enzyme activity, up to an optimum or maximum temperature. In mammals, this is around 37 degrees centigrade. At higher temperatures, the enzyme's protein is denatured. This results in the active sites changing shape and gradually declining in activity. Three. pH. Enzymes usually work efficiently within a narrow pH range. The graph shows that the enzyme works best within a pH range between 6.5 and 7.5. In greater acidity or alkalinity, the enzyme will be denatured. 4. Enzyme concentration. As long as both temperature and pH are at optimum values and there is a high level of substrate, the rate of reaction will increase as the enzyme concentration increases. Five. Substrate concentration. If the concentration of the enzyme remains the same, then a maximum rate of reaction will not be exceeded, however much the substrate concentration is increased. This is because the active site becomes saturated. Enzymes that work within cells, for example glucose 6-phosphatase, are called intracellular enzymes. Those that work outside cells, such as salivary amylase, are called extracellular enzymes.